Bismillahirrahmanirrahim. Assalamu alaikum students. Today we will start our first chapter, that is cell structure and functions. So to study cell structure and functions we use some techniques, and today we are going to study 5 techniques, and then in the next lecture we will study the rest of the techniques. So what are the techniques used in cell biology? We will discuss one by one. The first technique is cell fractionation. Now, how can we define cell fractionation? That it is a combination of various methods used to separate a cell organelle and components based upon size and density. So why do we use it? Actually, if you want to study a cell, you have to study it under a microscope, electron microscope, of course. Then remember one thing more, that the principle of cell fractionation consists of 2 steps. Number 1, homogenization, and number 2, centrifugation. Let's see them one by one. The first step is homogenization. It is the formation of a homogeneous mass of cells, which is also known as cell homogenate or cell suspension. It involves the grinding of cells in a suitable medium with correct pH, ionic composition, temperature, and the presence of certain enzymes that can break the cementing substance of the cells. When we are talking about, if we are talking about the plant cells, you can see here that these cells are joined together in the form of a tissue, and here you can see many cells which are joined together, and here where the pointer is, you can see it, this thing, the middle lamella, this one, this, it is present between the cells and it acts as a cementing substance and it joins the cells together. So if we want to separate the cells from each other to make a cell homogenate, what we have to do, we have to break this so that the cells can separate from each other. So for this purpose we use pectinase, because this middle lamella, this thing which is a cementing substance, it is made up of pectin, and to break it we need an enzyme, that is pectinase, and we do it in a homogenizer. What you are looking at is a homogenizer. It is more like a food mixture or blender, and when we mix the cells in it and operate it, we get a uniform mixture of cells which is known as cell homogenate. And after this, after homogenization, our next step comes, that is centrifugation. So the mixture from homogenization is centrifuged in a centrifuge machine. So what are you looking at is a centrifuge. Now, what is centrifugation? It is the process to separate substances on the basis of their size and densities under the influence of a centrifugal force. For this purpose, we use a machine, what you are looking at, a centrifuge. Now, this machine actually, you can see this is the machine, and in it we put test tubes. They are just like test tubes. And the tubes contain the contents which we want to separate. Now, here you can see inside of the centrifuge machine. Here it is, you can see this one, this is rotor which will move, and when it moves, it will exert a centrifugal force on the test tubes here in this here and here, and there are also many others in which we put our test tubes. So we will put our test tubes here, and also there are other points, and when this rotor operates, it will exert a centrifugal force on the tubes. So when the tubes spin, the centrifugal force acts on them and the contents are separated. Now actually there are two major ways of centrifugation. This is how we can do centrifugation, and number one is density gradient centrifugation and the second one is differential centrifugation. Let's study them one by one. This is what we call density gradient centrifugation. Now in it, the cell components of different sizes and densities are separated in different layers or sediments. Layers are also called sediments in tubes containing ionic medium according to their size and densities. So you can see here, here you can see we are doing density gradient centrifugation and we separate them in layers. So this is a tube and we put it in centrifuge, and what we get, you can see here, these particles, these and these are the same. So they are not. So we separate them according to size and densities into different layers. And what do you have in this is the upper sediment, small particles, and the largest one are in the bottom layer. So when we move from bottom, that is where the most dense and largest particles are present, we move towards the upper layers. They get smaller and smaller and smaller. The smallest ones are in the upper layer, the most upper layer. There is another picture, you can see it here. It is about the blood. So when you centrifuge it, what you get? Red blood cells, granulocytes, they are present in the medium, lymphocytes, and then plasma. So these are the layers we will obtain. So this is why we call it density gradient centrifugation, and in it we separate the parts into different layers or sediments. The second one is differential centrifugation, and you have to remember certain things about it, that when we talk about differential centrifugation, you have to remember the sedimentation rate. Actually, every particle has a sedimentation rate. So if you are talking about a particle of a given size and shape, it will have its own sedimentation rate, and on it depends how fast will it settle in the sediment. Now the faster the rotation of the centrifuge, the smaller particles will sediment. Now what we do in it, that we actually have a series of increasing speeds, and at each step, the contents which make the sediment in the bottom of the tube are called pellet. Here you can see, here we start, and here this one which settles, this is pellet, and above it portion which did not settle, this is supernatant. What we will do, we will put this supernatant into the next tube, and hence we will have one pellet. This is pellet. We have separated one pellet. Then what we do? We increase its speed, and what will happen? We will have another pellet here, and again the part which is not sedimented, it is supernatant. Then we put it into the next tube. What will happen? We will increase the speed once again, and after each speed we keep doing that we draw off the supernatant and centrifuge again. So here you get another pellet, and in the last these particles are left in the supernatant, they will also separate. So what you see, you have different pellets. This is pellet 1, pellet 2, pellet 3 and pellet 4. So we have separated all the particles. The next technique is differential staining. Actually, what happens that most biological structures are transparent. Now, if you want to see them, we have to color them. We use certain dyes so that they must be differentiated. So to differentiate the parts, we have to apply certain dyes, and the techniques in which we use these dyes is known as staining techniques. So these techniques are known as staining techniques. Now there are two ways to do that. When we use, number one, single staining. When we use only one stain, for example, borax carmine to stain the nucleus, it is known as single staining. The second one is double staining or differential staining. In this technique, we use two stains. One that will stain the nucleus and the other which will stain the cytoplasm. For example, hematoxylin is used for nucleus and eosin for cytoplasm. So here you can see double staining and how it helps you to differentiate the structures. Here you can see that you can differentiate the parts of the cell. So it becomes easier for you to study the cells when you use two dyes or double staining or differential staining. So it will help you to do that. Then the next technique is micro dissections. Now actually in it a variety of techniques are used, and in this method we use a microscope to assist in dissection. Now what is its use? We use it to remove tumors or granules from delicate tissues or cells. Delicate tissues or cells, for example, brain, heart, nerve cells. Now, in this technique, the image is seen on a large TV screen or monitor while dissecting. Different types of micro dissection are, or you can say the different kinds of techniques involved in micro dissection are: number one, chromosomal micro dissection; number two, laser micro dissection. Let's see them one by one. Number one, chromosomal micro dissection. Now actually in it we use a fine glass needle. Here you can see the fine glass needle. This is the fine glass needle, and we use it uh, under a microscope to remove a portion from a complete chromosomes. For example, you can see here this is a complete chromosome, and we are using this needle to just uh, remove a portion from this complete chromosome. Then we have laser microdissection, and in it we use laser through a microscope to dissect selected cells. So we put here the selected cells, and a laser beam is used to cut the cells which we want to study. And here you can see there is a monitor present which will help us to locate those cells. Then the next technique is tissue culture. Now actually in tissue culture we grow cells or a tissue on chemically defined nutrient medium under sterile conditions, and we call it tissue culture. This technique can be employed for both plants and animals. So two types: it can be plant tissue culture, it can be animal tissue culture. Let's see them one by one. This is showing you plant tissue culture. Can you see these small plants growing in these test tubes? These can be grown in test tubes, petri dishes, flask, with what you use in your laboratory, conical flask. So actually plant tissue culturing is used for plant cloning. And what is plant cloning? Production of genetically identical plants. And you know what are genetically identical plants known as? They are known as clones. Then the second one is animal tissue culture. Now in animal tissue culture, we use individual cells. We grow individual cells from a to form a single layer of cells over a surface of a glass container. Here you can see the surface of a glass container, and these cells are making a single layer on it. So this is what we do in this technique. Now there is one question, that why do we use it? So animal tissue cultures are used to see any abnormality in cells. For example, if you want to observe cancer cells or to see any chromosomal disorders in the cells, so you can use this technique. Then we have chromatography. So it is a technique which is used to separate different chemical compounds from a mixture. So you have a mixture and you want to separate the chemical compounds from it. We generally use it to separate the mixtures of proteins, amino acids, or photosynthetic pigments. Now there are different types of chromatographic techniques. Mostly used technique is paper chromatography because it is simple and the most widely used, and it consists of two phases. Here you can see the mobile phase. This is mobile phase, and this one, which is a filter paper, or we also call it chromatogram, this is known as the stationary phase. So there are two phases, stationary phase and mobile phase. Now mobile phase actually consists of a solvent and in it a mixture. Here you can see this is the solvent, which is our mobile phase, and in it we dissolve the sample mixture. Now what happens that it passes through the stationary phase. Here you can see it is passing this solvent front. This is known as solvent front, where it is passing. So when it passes through the stationary phase, what happens that the molecules of mixture sample begin to separate as dots at different places on the stationary phase according to their individual affinity. Then paper is sprayed. We take out this paper actually after that, and we spray it with a liquid locating agent, which is a staining dye. Mostly ninhydrin is used for this purpose. So when we spray it with the liquid locating agent, it shows up the dots as colors, which we can see. So, and this, what is this? Known as paper chromatogram. And this whole apparatus, this is known as chromatography chamber. So that's all for today, students. Thanks a lot. Allah Hafiz.